Okay, so I will talk about the ethical issues related to intervention trial, which is the most fascinating part of research for any clinician. We always want to do trial, but it contains lots of nitty-gritty related to research ethics. So we should know before we start any trial. So as a disclaimer, whatever I'm talking is based on my experience and little bit of teaching, but not any formal training. And just a simple word before you think about any question in your ethics related to trial, that will you be happy if it is done with you in a trial? If your answer is yes, then 99% chance is you are correct in your trial. If you are a participant in that trial and that thing happens with you and you are happy with that decision, then you are 99% correct. This is the simplest litmus test for your ethics integrity in any clinical trial. So these are the content I will talk briefly about one by one. What is an intervention trial? Trial word is not so simple. Any research study that prospectively assign human participant to one or more health-related intervention, right? To evaluate the effect on health outcome. Please emphasize health outcome could be anything. It is not that always you are trying to reduce the pain or agony of the patient. If even you are trying to improve the quality of life, that is also an intervention. If you are trying to reduce the financial burden on the patient without any physical benefit, that is also an intervention. If you are just reducing the discomfort, that is also an intervention, and that is also a trial. So many times we think that we are asking only question about quality of life with the patient, so it is not a trial. If you are prospectively assigning a participant in two groups, that in one group I will ask this quality of life questionnaire and second group I will not ask, that is also a trial. As easy to understand. Then participants are assigned to group by the researcher. This is the problem that where the difference between the standard of care and the trial comes in, where the patient is losing his or her choice to decide about the treatment option. Even the two treatment option may be completely equivalent in context of outcome, finances, pain, everything. Even then, patient has lost his choice to choose out of the two options. It is you who are going to choose the option. Then it becomes a trial. That is the crux. So phases of clinical trial for ease of understanding. Most of the time what we do, it is phase 3 trial. We are hardly doing anything phase 1, phase 0, phase 1, 2 or 4. So what are the phases? Phase 0 is the earliest. Whenever new molecule is discovered, identified, we try to do basically understand the distribution and metabolism of the drug. So very few numbers, very few participants are done, and most of the time this study is done in ICU setting in healthy population, right? So it is a part of regulatory requirement. That is the phase 0. Then phase 1, the primary objective of phase 1 study is to study the pharmacodynamics and pharmacokinetics. We are not expecting any benefit out of the drug, and that is why we are doing it in healthy population. We don't know how this drug will behave, what pharmacodynamic or kinetic effect will have. It may activate some of the receptor in the human body which can take life. It may block some nervous system and can cause palsy or heart attack, anything. So all these trial phase 0, phase 1 are always done in ICU setting, right? And healthy participant, very few numbers, as minimum as which can just fulfill your hypothesis and give you reliable data on pharmacokinetics and dynamics. That is good enough, usually 8 to 10 participants. Phase 2 is explore the therapeutic efficacy. Please mind, it is the exploring the therapeutic efficacy, but it is not the comparing therapeutic efficacy with gold standard. Where you are comparing with gold standard, then it becomes phase 3 trial. You are just seeing whether this drug going to have any beneficial effect for a certain disease to a patient or not. So you will include deceased person, not the healthy person here, because you want to see the benefit effect of the drug, right? So relatively larger number because the safety is more or less established in phase 1 trial. You might like to see some minor or intermediate severe side effect or some uncommon major side effect. Major means less than 1%. So that is why you take 100 to 200 participant, and it can be done in hospital setting. Phase 3 is to confirm the therapeutic benefit. Means here, most of the time, what studies we are doing, comparing one drug with other, one dose with another, one route with another route, one duration of treatment with another duration of treatment. All these are phase 3 trials. And we are comparing with the gold standard. So we should know what is the gold standard or standard of care for a given treatment. So typical RCT is clinically what we do it is. And we might be doing it for a new indication, new route, new dose, new dosage form, new preparation, or new combination. New indication itself is coming in phase 3 trial because the drug safety, safety, metabolism, pharmacodynamic, pharmacokinetic effect has already been established in phase 0, 1 and 2 phases. That's why we are directly going into phase 3 trials. Like we have used lots of ivermectin during COVID. So that was all phase 3 trial. Phase 4 is to post-marketing surveillance. We want to see very, very uncommon side effect, some side effect which occur in population, means in large number, which led to withdrawal of some of the drugs. We all know. So these are usually not done in clinical setting. We are not doing it. It is done by the industry. Type of intervention trial, it could be preventive. We are trying to prevent a disease like vaccine trial. You are giving vaccine to prevent a disease. Diagnostic, you are comparing ultrasound versus CT scan or any other intervention, any other diagnostic modality, or it can be therapeutic, which is most commonly we are doing as clinician. So intervention could be in form of either medication, psychological, means counseling. Counseling means you are counseling one group of participant about the diet therapy with a dedicated dietitian, and another group you are giving standard of care, just clinician is giving counseling. That is also an intervention. That is also a trial. You are not doing any harm. You are giving benefit to the patient, no financial burden to the patient. In spite of that, that is a psychological intervention. So it can be health education in form of health education, radiation safety, vaccine, surgical device, technique, drug, anything it could be. It can be established intervention, but not substantiated by scientific evidence. Or established intervention for a new purpose, new purposing of the drug, as we told. So anything could be an intervention. This will also be an intervention if you are withholding an established intervention. Some standard of care is there, but you are not giving a patient, you think that it is useless, that is also an intervention. Means it is a negative intervention, but still it is an intervention. Altered administration means change in route, dose, duration, form of drug. Earlier the drug was given in tablet form. Now you want to compare the syrup form with the tablet form. That is also an intervention. Common in pediatric practices. Change in method of delivering care, such as DOT, means earlier it was telling that patient you can take the medicine and eat at home, and in DOTS you are telling you come to the hospital and eat. That is also an intervention. So you should keep your eyes open and start thinking that intervention can be anything. And trial is anything wherever patient is losing its right to choose the intervention. You are choosing the intervention and you are randomly assigning into one of the arm. So there are two types of trial. One is academic trial and non-academic trial. Non-academic trial is done by the industry, and these are needed for regulatory approval of the drugs. So for drug approval, new drug intervention, basically we cannot do in a medical institution. We can do as a center for a pharma industry, but we cannot do ourselves. If we are doing ourselves for our research, usually what we do for our thesis or publication purpose, that is a academic trial. So a systematic study of new drug in human subjects. So new drug, not the new route, dose, duration, but new drug. Even not the new indication. That will be an academic trial. So that was the difference I have differentiated. In academic trial, we should be very cautious because investigator has two responsibility. One responsibility, you have to work as an investigator toward ethics committee, toward patient, you have to give all the honest data and etc. And also you have responsibility of sponsor. Means if any untoward action happens, severe adverse event, then the compensation has to be given by the investigator. Because in clinical trial, academic trial, all the responsibility lies with the person who is doing trial. Here, the condition may be different because here sponsor is institute. But still we have limitation. We don't have much of mechanism to pay compensation. And that is why sometimes we are conservative in doing trial where the risk is likely to be there. So financial arrangement has to be made. A blanket insurance is to be taken either by the person himself or by the funding agency who has sponsored the trial or by the institute to bear the cost related to severe adverse event of the participant. Before we start any trial, we plan any trial, these are the some important points which I want to emphasize, which we commonly miss. First is equipoise of evidence. Anybody can tell me what is equipoise of evidence? If there is no equipoise of evidence, then ideally we should not start a trial. It is totally unethical to do a trial if there is not equipoise of evidence. Then what is equipoise of evidence? Yes, means result is not conclusive based on the previous data. Many times what we do, just to get our project passed, we write there is equipoise of evidence and we quote one positive study and one negative study and say that, but we should be clear in our conscience that really there are data to support and against equally, or we are hiding the truth with the ethics committee. Because even if you are getting ethics committee clearance, later on responsibility lies with you because ethics committee is giving you clearance based on the data you have provided to the ethics committee. If ethics committee set up another committee and review that what literature was available at that time when you submitted the project, then responsibility is yours because you have not given the correct information to the ethics committee. So we should be very cautious. Just to get our project sail through, we should not try to create an equipoise. That is an important point. Second is assumption of sample size calculation. We are all running a race of 3 years. Extramural, intramural, DM course, everything, 6 months is gone, then clearance, start project. So we have 1.5 year, 2 year of duration to do a project. So we want that we should recruit all the patient in 2 years. This point is more important for the faculty, I believe, that we should give a project which is feasible within the given duration of time instead of adjusting the sample size for the duration of time. This is not acceptable because for any study, you can calculate a sample size of 50 is good enough or sample size of 50,000 is inadequate. It all depends what you have assumed. Sample size is not a sacrosanct thing. It is a what calculation, what number you have put in for assumption and your calculation. If you have assumed wrong, then your sample size will be wrong. So sample size calculation should be justifiable, should be ethical, but it should be feasible. But just to make the study feasible, sample size should not be adjusted. This is an important point. Whenever you do a study, please take care of it. Because another point, what happens, you might get rid of ethics committee, funding agency, you will write paper, but when you will send it for review, then paper will be rejected that your sample size calculation was not based on appropriate selection criteria. It was not appropriate criteria. Because reviewer will also check your sample size based on your assumption, right? So ethic approval before the start of trial, at least for interventional trial, where you feel we are doing some intervention means, see, here I will little bit laxity if you are doing something like counseling sort of thing. Ideally, you should start any study after getting ethic clearance, but you know you are not going to do any harm, it's not possible to the patient, then you can still consider to start your study and later on in due course of time, next 1 month, 2 months, by the time committee meeting is happening, you can consider to take clearance. But if you are using some drug which is likely to have even minor side effect, please don't start your trial without getting ethic approval. Ethic approval means ethic approval. It is not the ethics comment. Many times we are getting ethics comment that modifications are suggested, please resubmit. Don't take it as approval. That is only comment. Approval means when the final approval is given. And when you are planning a trial, try that there should not be any additional financial burden related to travel. Means patient should not need any extra visit for your trial, any extra stay, because it will cost money. It will cost loss of wages for the family member and the patient. I know practically it is not possible many times. So at least we should try to minimize it as much as it is possible and avoid unnecessary investigation. When we plan a trial, we will, every visit we will write hemogram, LFT, PT, creatinine, but really do we need it for our analysis? If we don't need it, then why to investigate it? Because it will cost money and unnecessary wastage of time and stay and wages, etc. So we should be considerate about the expenses related to our research project. Use of placebo. When we are choosing a placebo, then in which trial, which type of trial we can use a placebo? When there is no established effective therapy available as a standard of care, then we can use a placebo. When we know that withholding the standard of care will not adversely affect the outcome, then you can use placebo. Or disease is self-limited, means no drug is going to give any benefit, then you can give placebo to improve the quality of your study. Or use as a comparator would not yield scientific reliable result. If you feel that if you are not comparing your intervention with placebo, your result is not likely to be reliable. It is not that result is going to be significant, it is reliable. Then you should use placebo. Because a study is not done to get a significant result. Even negative study should be as much welcomed as a positive study. So ethical aspect of a multi-center trial, every center has to take a clearance from its own center, and the PI from that center is responsible for recruitment and ethic clearance and ethics associated issues in his or her center. It is not the PI. That is important. So if I am doing a multi-centric trial and 5 centers are there, so every center will take their own ethic clearance and they will be responsible for any unethical thing which happens in their center. It's not me. And so they have to start a study in their center after securing their ethic clearance. It is not that the PI has got the ethic clearance and we can start the study. It is not. Every center has to take it. Safety monitoring. Safety means safety of the participant, safety of the investigator, because after all, safety of the investigation is also important, and safety of the document, data, everything. So trial related documents should be like signed consent form must be taken, PID must be signed, and they have to store for 7 years at least for any trial. DCGI approval must be taken for use of any new drug. Obviously, new drug, if you are doing new indication, then you don't need any approval. If you are using an altogether new drug, then it has to be a pharma industry started trial. Then please check that they have taken DCGI approval for the trial. And approval of the Animal Ethics Committee, if your study involve animals in any form. Approval from Institute Biosafety Committee, if you have involvement or use of any biohazard material, which can pollute the environment or can be dangerous for the research staff or the hospital environment. Biosafety laboratory should be there depending upon the requirement of your research project, and radiation safety measures should also be taken, particularly in radiology or nuclear medicine related intervention trial, because the risk of radiation. And safe disposal of biomedical waste means the remaining sample, tissue sample, blood sample should be discarded in a proper manner and not simply in a dustbin, which can contaminate the environment and can be the source of epidemic of any infectious disease. So data safety and monitoring board, this is one thing which is needed. If you're doing a trial, it is not in our institute, but if you have data safety monitoring board and your trial is monitored by data safety monitoring board, your quality of your study improves, reliability of your data improves, and obviously the chances of getting publication in a high impact journal improves markedly. But this is not in your hand, it is the institute policy. Definitely the kind of studies which must be covered under data safety monitoring board. So it is not a mandatory thing that every trial should be covered at a DCMB, but the trials which are related to saving the life means where the outcome is very positive, so that the result should be reliable. Just to account for the reliability, accuracy of the result, the data should be monitored by some independent board. So same thing, what kind of intervention should be monitored? So I will not spend time on it because it is limited use here. Then research related injury. Harm or loss that occur to an individual as a result of participation in research, irrespective of the manner in which it has occurred. It include both expected and unexpected adverse event. Means you are using a drug for seizure disorder and patient develop cardiac arrest. Even then, it may be related to the drug. It is not related to nervous system, but still it may be related to the drug. So related or unrelated event, both. Serious adverse event related to intervention or any medical injury caused by the procedure. Serious adverse event, what is the definition? These are the conditions which are counted as a serious adverse event. And for any trial, a serious adverse event has to be reported by any means, in writing, by email, by phone, within 24 hours of its occurrence to the Institute Ethics Committee. And within 14 days, a detailed report has to be submitted. Yeah, same thing. And as we learned earlier, that we should have some mechanism to cover up the serious adverse event, so that the financial burden is not coming to the participant. And relevant, whatever is appropriate by law, the compensation should be given to the patient or their kin. These are the regulatory requirements for some specific type of research, like National Apex Committee for Stem Cell Research and Therapy. So if you are doing any trial related to stem cell, the study has to be approved by Institute Committee as well as by these central committees, not only from the Institute Committee. Genomic data deposition has to be done in repository. Even the study is not funded by any other funding agency. It is completely funded by the institute, but all the genomic data has to be deposited by law in repository. And we have guidelines related to good clinical practice, laboratory practice, manufacturing practice, IUs, GCP. So related to which guideline is working with your trial, you need to know. Means if you are doing some trial, then we have guideline that drug which you have used should have been manufactured in a factory which is approved by good clinical practice guideline. Same way, if you are doing a trial, then investigation should be done in a lab which is NABL or NABH accredited, so that your result is reliable. Interim analysis is done many times, but not in all the studies. It is planned beforehand and result has to be communicated. If you have decided that you will do it, then you do it and communicate the result, get a permission from ethics committee, and then go ahead with the continuation of the trial. And it is done basically to avoid unnecessary harm to the participant in the intervention arm. Harm in the intervention arm, if your intervention is harmful. To avoid unnecessary loss to the participant in the control arm, if your intervention is useful, because you are dividing your control arm from the intervention, and intervention is fruitful. That is why. And result must be communicated. Another thing which in last I want to say that whenever we are doing trial, just for the sake of publication, use of fake data, data duplication, data fabrication is very common practice. It may bring a small publication for you, but it can bring a very bad name for the country, for the institute, not for you. It will not bring any bad name for you. You will be only benefited, because at the end, when something is blacklisted, it is not the person, it is the institute. यार वहां के लोग तो फर्जी डाटा भेजते रहते हैं, हम भी आपस में बात करते हैं ना कि वहां का डेटा तो रिलायबल नहीं होता, वो फर्जी भेजते हैं, हम किसी एक की बात नहीं करते. सो हम अगर कोई रिसर्च प्रोजेक्ट में डेटा फेक करते हैं तो उसका इम्पैक्ट पूरे इंस्टीट्यूट पे, पूरे कंट्री पे आता है. सो बिफोर डूइंग एनी फैब्रिकेशन, एनी फैल्सिफिकेशन, we should be cautious that the impact may be many-fold than what we are seeing. It may be 1,000-fold than what we are gaining just by a paper. And at the end, for any trial publication requirement, when you submit it, the end point of any trial for us is PubMed. That CTI registration is must. You might have registered in clinical trial, but even then CTI registration is must. So if you want to register only in one registry, then it should be CTRI. If you can register in two, then you can go for two. But please be sure that you are registering the same thing in two places. It's not that in one registry you are writing 60 participant, in another 66 participant. Then it will be very, very dangerous. So, and follow your reporting guideline in context of trial. When you are publishing the protocol, you have a standard protocol publication guideline in consort. You have a standard publication guideline for trial, observational studies, systematic review. So please try to follow those guidelines. Role of funder in a study, they will ask. Most of the journal, when they will publish the paper, they will ask you. So you should be clear what is their role. Means they have just provided you the consumable items. They have provided you manpower. Do they had access to the randomization? Do they had access to the participant data? Where the investigation was done? Means you have just collected the sample and send it to their lab for investigation. That means they can manipulate the result. That is what journal wanted to know. To what all the funder had access to. Means how much the funding agency or the funding company had power to change your result. Conflict of interest, obviously we have to give, and trials supervision by the Data Safety Monitoring Board if possible. In summary, participants are assigned to a group by the researcher. This is a trial, right? This intervention can be anything. And we should be honest while planning the sample size, comparator, equipoise, financial burden on the participant, and do not forget to register in CTRI. Responsibilities are increased when you are doing as an academic trial, because then the budget is limited, responsibilities are more. And think of impact of any dishonesty done by us for a small trivial benefit. Thank you.